All right, I'm going to do my best and talk as loud as I can, but you'll get the recording off of the video. Okay, so we'll pick up where we left off 2 classes ago, essentially. We were discussing replication, right? So we talked about DNA and the chromosomes, and we talked about replication, and then some of the technologies that have come out of what we've learned about replication. And so we talked about restriction enzymes, cloning, and some sequencing technologies. So just a review question from last time, if anybody can shout out the answer. I heard 2 different answers. I heard an A and a C. C? Okay, so can anybody tell me what does DNA A do? Okay, can anybody tell me what DNA A does? Yep. Okay, so then so we're going based on answer C, right? So what does the primase then do? And then DNA pol 3. Yep, so that actually does the DNA replication, right? And then why do we need the ligase?
Combine the ligase.
Yep. Yeah, so it's to close up the gaps for the lagging strand, right? For the Okazaki fragments, right?
I know.
Okay, good. So today we're going to go into transcription and translation, and as you can see, there's a lot to know about transcription and translation. So we'll go through it, but some of the main ideas is you want to know what enzymes and what components are responsible for each process, how the different processes work. We'll also talk about there are a lot of antibiotics out there that inhibit both processes. And then this chapter also talks about protein modifications and what happens post-translationally. So we'll talk a little bit about that too. All right, so hopefully everybody recognizes this, right? The central dogma. So it's how we go from DNA to mRNA to protein, right? So the process that goes from DNA to RNA, that's transcription. And so the main player that's responsible for this is RNA polymerase. And then for translation, it goes from mRNA to protein. Again, the main player in that is the ribosome. So hopefully this is not new to anybody, and we will start with transcription. So as I mentioned, so RNA polymerase is what is responsible for performing transcription. So it is a DNA-dependent RNA polymerase, meaning what comes first is DNA is what it's reading, right? And then RNA polymerase it's making RNA, right? So this is when we talked about viruses, right? We had talked about RNA-dependent RNA polymerases, right? Because they were reading the RNA viruses and making RNA copies. So in this case, we usually just refer to RNA polymerase as just RNA polymerase in this case, and this is like our standard reference. Okay. So what you'll also hear about is the RNA polymerase holoenzyme. So this is actually the core polymerase plus a sigma factor. And so that whole thing combined is what we refer to as the holoenzyme. The core polymerase, it has a whole bunch of subunits. So it has 2 alpha subunits, 1 beta subunit, a beta prime subunit, and then this omega subunit, which is not necessarily required for transcription, but it's there. So this is what they look like. So this is just the core polymerase and what it looks like when it's assembled. So you can see, and in here, this is where the beta prime, and this is where the DNA actually goes through. And so the beta prime subunit is actually where the catalytic site is for putting together the RNA strands. All right, so sigma factors. So sigma factors are important because this is the part that actually tells the polymerase, like, where do I start? And this is what helps it find the promoter sequence. So transcription always starts at a specific DNA sequence. And so one sigma factor can recognize several somewhat similar sequences. And so the sequence doesn't have to be identical, but there will be some characteristic bases that are similar across all those bases that it will allow it to recognize that base. So in E. coli, what we call the housekeeping sigma factor, so the one that kind of does most of the genes, is called RPOD, or more commonly known as sigma 70. And there are several other sigma factors that control other genes, but as you can imagine, they are only, it uses as a regulation technique so that you only make these certain genes in the cell when it's necessary. So some of the other cases are in heat shock, in cases of chemotaxis or motility, right? We had talked about that the cells don't actually make the flagella unless they need to, right? And so then you also have ones for RPOS, which does the stationary phase or stress response. And then there are some cases for the nitrogen metabolism in some bacteria. So again, all of these will recognize different sets of the promoter sequences. Going back to this slide, so here for the different sigma factors, you see that there are different consensus sequences for the -10 and the -35.
Do we need to know what all those sigma factors do? Like every single, like each of those sigma factors?
No, just know that there are different ones, and why would you need different ones? So the promoter is the sequence in the DNA. So this recognizes the start of transcription, which is noted. So there needs to be a way for us to count nucleotides. And so we always call the first nucleotide that gets transcribed the +1 nucleotide. And then so the promoter sequence, so relative to +1, the promoter sequence is generally, so, oh, this is missing a word. So this is generally centered. So it's generally centered at the -10 and the -35 nucleotides. So if this is the +1, so these are a bunch of different promoter sequences that are aligned. And this is the +1 site. And then so you go the -10 and then the -35 region. So these are the regions that the sigma factor are really recognizing. And so what you can do, and what people have done, is basically taken all of these promoter regions from all the different genes that say sigma 70 regulates, and then align them, and then you can get what you call these consensus sequences. So in general, the ones highlighted are the ones that are most often the same within that sequence if you align them all. And so that consensus sequence is the one that's most recognized by the sigma factor. And okay, so this is just again another picture of what the whole thing looks like with the sigma factor in there. And so what happens is the sigma factor is actually going to kind of go along the DNA and scan the DNA for the sequences that it's looking for. And what allows that to happen is, remember we talked about the structure of the double helix, and it has the major and the minor grooves. So that's actually what allows it to be able to fit in there and be able to actually see what the nucleotides are in the DNA. And this will become a little bit more clear just in a couple of slides, but what this also does is it shows you a picture of what the transcription bubble looks like. So as you imagine, in order to transcribe, you actually have to unwind the DNA, right, to get both strands open up so that it can start transcribing to the RNA. So that's what, it doesn't like rip the whole thing open all at once, and so it creates what's called this transcription bubble, where it has just this short span that's open at one time, and that kind of just moves along as it's going. All right, so transcription has 3 different stages: initiation, elongation, and termination. So in the initiation stage, RNA polymerase binds and it melts the DNA, and then it catalyzes the placement of the first RNA molecule. In elongation, you just keep getting the addition of each new nucleotide. And then in termination, there are specific sequences that actually that result in the ending of transcription, and we'll talk about those sequences specifically. All right, so first for initiation, So as I was mentioning, the polymerase, so RNAP stands for RNA polymerase. The holoenzyme, so the whole thing put together, the sigma factor, it essentially goes along and it scans and it looks for the appropriate promoter sequence. So once it binds to the promoter sequence, it forms what's called the closed complex. And then at this point, it starts to unwind the DNA. So it'll start unwinding the DNA, and that essentially what begins to form the open complex. And typically, you'll find that transcription will start with a purine. I'm not really sure why that is, but just in general, it starts with an A or a G. And then as transcription gets going, after about 9 bases or so, the sigma factor will release, and that can go on and bind to another RNA polymerase and be used to find another transcription start site.
So that's the start site, or is that something?
Sorry, what?
The start site, is that where it's...
So the start site is going to be, let's say, I don't know how this is counting. This is not really proportional in this picture, but let's say it's up here, right? So it's going to be here if this is +1. So it starts at +1. So yeah, so that's where the first one goes. It has to do with the fact that the transcription bubble was a certain size, and so it's like, I think it's 17 nucleotides or something like that, and so where the location is, and that's where it'll start. Okay, so for elongation, it's pretty simple. It's just, oh yeah, go ahead.
About the last slide, so as soon as it starts unwinding.
Okay, so from here, it's pretty simple. It's just adding new nucleotides, right? Just reading the template strand and just adding new nucleotides, new RNA nucleotides. And so it does this at a rate of about 45 bases per second. So that's really fast, right? And we'll try to remember that number when we talk about translation for a couple of reasons. So as always, when you're creating bonds, you usually require some kind of energy. And in this case, the energy actually comes from the base itself. And so if this is the growing RNA chain, you are going to add it to this phosphate here, and so this release of this molecule, the pyrophosphate, and so that's the energy that is used in creating these bonds. All right, so for termination, for bacteria, there are 2 main styles of termination. The first one we'll go over is the rho-dependent termination. So Rho is another protein. It's a hexameric protein, so that can bind. It'll generally bind to C-rich sequences in the open reading frame. So open reading frame meaning that's what's open for translation, right? So it'll generally bind this C-rich sequence. The key is that this Rho is also an ATPase, so it uses ATP to essentially shove itself back up the sequence. So it'll go 5' to 3'. It'll attach on this C-rich sequence, and then it'll use energy to make its way back closer to the polymerase. In the meantime, the RNA polymerase will pause at a specific pause site in the sequence, and essentially it's almost like it's waiting for Rho to come along, and Rho is essentially going to go over there, and it's going to start unwinding the RNA DNA complex, and then that action will actually cause the whole thing to come apart, and so the polymerase will come off at that stage because it's melting the RNA DNA duplex that's being made. All right, the other style of termination is called Rho-independent termination, and another term used is the intrinsic termination. So this one occurs at a specific RNA structure, and so this is called a terminator sequence. You'll sometimes hear they're called intrinsic terminators because it goes with intrinsic termination. So what it is, it's a hairpin loop with a GC-rich stem. So a hairpin loop, that's what this structure is here. This is what we call a hairpin loop in RNA structure terminology. So the GC-rich stem, meaning these bases here are high in GC content. That doesn't mean that it's all GC. There could be A's, but it's going to be high in GC. And then what it's followed by right here is this poly-U tract. So this is important for several reasons. So the poly-U, so when this is being made, this UA DNA hybrid is actually very unstable. So remember, the GC bonds are stronger, right, than the UA bonds. And so this is pretty unstable. You'll have this tract of U's, and you can get, you know, about like 6 to 9 or 12 or some odd number of U's. So it creates this unstable region. The polymerase will actually pause here at the poly-U site also. And then basically what's happening behind it, so your polymerase is going along, transcribing the RNA. The RNA is coming out. You imagine that the RNA is coming out the back of the polymerase, but that's going to be folding while it's happening. So the RNA is folding, and it has this sequence here to form this hairpin. So there's lots of G's and C's here, so it wants to form those bonds there. So as these things come together, so that is a very strong bond formation, and then essentially this is unstable, and as the hairpin comes together, it essentially kicks off the polymerase because it's not stable on the U tract, and then this other structure is being formed, and it essentially just like ratchets and it falls off. So the other protein that's involved is this NUS-A. So this actually assists with two things. It assists with the pausing at that U tract, and then also assists in the hairpin formation. Any questions about termination? So it's always going 5' to 3', so it's going, you know, well, yeah, so 5' to 3' is here. All right, so like we mentioned, there are antibiotics that block transcription. The main one that your book talks about is this rifamycin B. So this will actually bind to the beta prime subunit, so near where that active site was, and it actually essentially just binds there, and it blocks that channel. So it blocks anything from happening. All right, so we'll move on to translation. So now what we want to do is go from this mRNA to a protein, and we have to essentially change languages, right? So we have to go from RNA nucleotides to amino acids. So again, hopefully this is not new to you, but so it's basically the decoding of the RNA message to synthesize proteins. And so what we have are these codons, which are triplicate nucleotides. So there are specific codons that correspond to a specific amino acid, and this is basically the table that allows you to translate that code. What you'll notice is that there are several codon sequences that can apply for the same amino acid, right? So that ability is referred to as being degenerate. So there are more than one code that corresponds to a single amino acid. And then there are some stop codons here. So these actually don't correspond to an actual amino acid, right? So this is the signal to tell it that that's the end of the protein. Green. Oh, that was just to point out, so most of these, if you look at them, they're kind of all in the same area, right? So usually what happens, it's like the first two will be the same, and then the third one will be different, and all of those will be for the same amino acid. In this case, they're just trying to highlight to show you that there are some serines over here that start with UC, but then there's also the serine over here that starts with AG. And so that the same thing for the green one, the arginine. Yep.
Sorry, what could the blue and green colors represent?
Blue and... Okay, so when it comes down to translation, you know, we talked about the ribosomes, the main thing that's actually doing the translating, but in reality, the tRNAs are playing a huge role because they are actually the ones that are decoding the messages. And so the so tRNA, it has a very specific, so tRNA is a completely an RNA molecule. It has a very specific structure, and so this structure here is what it looks like. That's what we call the secondary structure for this RNA. So if you, in 2 dimensions, what it would look like if it was flat. And so what this does is it has, so let's say, so there's 4 main regions, right? So it has each one of these regions here we call loops. And so the kind of major ones are the anti-codon loops, so that it actually has an RNA sequence that's available at that loop, and that is going to be complementary to the codon, right? So that's how it actually interacts and matches to what's in your mRNA transcript. The end opposite to that is where the amino acid binds, right? So that's what we call the acceptor end, so that's where the amino acid attaches. And these 2 other side loops here, they're actually used for recognition for this next molecule that we'll talk about is the amino acyl tRNA synthetases. So these are the guys that actually come in and attach the amino acids. So you have tRNAs floating around, but they need something to come and put the amino acid on them. And so that's what the amino acyl tRNA synthetases do. Generally, there's 1 per amino acid for these, and then so in order for that specificity to make sure that there's no errors in that, it actually goes and it recognizes all 3 of these loops and makes sure that those are the right ones, so it's putting the amino acid on the right tRNA. All right, so now we'll talk about the ribosome. So the ribosome is a combination of both proteins and RNA. It's generally, you'll always hear that it's broken up into the small subunit and the large subunit. So in bacteria, that's the 30S and the 50S. So the full ribosome is a 70S, where the S stands for Svedberg units, and that's based on sedimentation rates when you centrifuge them. So it doesn't go 30 plus 50 equals 80, just because that's not how the whole thing will sediment when it's being centrifuged. All right, so for the small subunits, so for the 30S, we have 21 ribosomal proteins, and the 16S are RNA. So everybody remembers what the 16S was for, right? Yes? Anybody?
Termination archaea.
Sorry, what?
To differentiate archaea from eukaryotes?
It's actually to differentiate anything, right? Every species. So yeah, so we have 16, all your cells have 16S, right? So that's what that's for. And then the 50S, so again, another set of proteins, so 31 proteins in this time. It has 2 different RNAs. So the 5S RNA and the 23S RNA. The 23S is where all the action happens in this, and so this is where the ribozyme activity is, and so that's what actually has the catalytic ability to do this peptidyltransferase. And then this is kind of just a picture to show you the units coming together and what the different, how the proteins fit around the RNA molecules. All right, so the ribosome has 3 different binding sites for the tRNA. So we have the E site, the P site, and the A site. The ribosome, so again, so just like for transcription, right, we need to know where to start translation. So instead of a promoter sequence, what we have is the ribosome binding site, or the Scheindel-Garneau sequence. So this is generally a purine-rich sequence of 4 to 8 bases, which is 4 to 8 bases upstream of the start codon. So we know that it's going to start translation at the AUG, at the start site, but generally it needs to, so what you can imagine, right, the ribosome, just because there's an AUG, if I start it here, but there could be another, like just 2 nucleotides down, there could be another thing that says AUG, right? So how does it know where the actual correct 3 start, right? Does that make sense? And so this ribosome binding site is actually what kind of guides it to the correct reading frame and to find the correct start, the correct AUG. Because remember, AUG also just corresponds to methionine, right? And so there could be other methionine amino needed in your protein, and so there are probably other AUGs elsewhere within the protein. Okay, so where was I? So there is there is a complementary sequence to the, so it's actually, so the shine dog RNA sequence is complementary to the 3' end of the 16S rRNA. So there is a part of the 16S RNA that is complementary to the A's and the G's that are in the shine dog RNA, and that actually will bind together, and so that's how it kind of finds that spot and locks itself in. And that will essentially, when those two bind, it essentially places the start codon in the P site. All right, so same thing for translation. There are 3 different stages, right? Initiation, elongation, and termination. So we'll talk about initiation first. So for translation initiation, there require these 3 different initiation factors. So for whatever reason, I don't know who named them, but they don't start with 1, they start with 3. So IF3 binds to the 30S. So remember, it has the 30S and the 50S, and they're together. But to get started, you actually need to separate them. If not, things don't actually make its way in there. So IF3 binds to the 30S. And then that actually allows the 2 subunits to separate. And then we have IF1 and the mRNA binding. So IF1 puts it into the A site, and that allows the RBS to kind of interact with that. So the 16S to interact with the shine dog RNA site, right? So this kind of allows that interaction to happen. Then IF2 is complex to GTP. So GTP, so the thing to remember about translation, the energy, so like we said, everything when you're doing these things requires energy, right? So a lot of these, so the energy in this process comes from GTP. So it comes into with attached GTP attached to IF2 with this FMET tRNA, and so that comes into the P site. So this, because remember the start codon AUG corresponds to methionine, and so that comes into here. So when that comes in, the GTP is hydrolyzed, and then IF1 and IF2 are released. And so when these guys get out of the way, that makes space for the 50S to come in and dock, and that now you have the full complex that's ready to go. All right, so for elongation, you have these basic 3 steps that are repeated over and over. So again, there are these other factors. So this time they're called elongation factors. So this EFTU and this EFG. So these again are going to be complexed with GTP to bring in energy. So in the first step, the aminoacyl tRNA binds to the A site. So so it's reading the transcript here. So the next codon will come in and it'll bind into the A site. So when that happens, the GTP is hydrolyzed, and so you get the EFTU with the GDP now is released. So this is now in the correct site, and what you have is the peptide bond form. So in the 23S catalytic site, that catalyzes the peptide bond formation. And so then now this peptide bond formation, actually what happens is it puts the growing peptide chain attached to the A site here. Okay, so the next thing you have to do is we basically want to move the whole thing over to the next codon, right? And so that's what we refer to as translocation. So what happens with that is we have this EFG comes in, and that's also attached to GTP. So that comes into the spot here and essentially just pushes everything over. And again, that requires the use of 1 GTP molecule there. So now everything's moved over, and eventually this part, the last one will exit out the E site, and then you can have your next one come in, and that'll just keep happening over and over until we hit the stop codon. So what I wanted to mention was, so this happens at about 16 amino acids per second. And if you kind of remember the transcription rate, anybody remember? Yeah, so that's about, so it's about 16 times 3, close, right? So basically, remember that transcription and translation are happening at the same time, but if one is going way too fast, so okay, let's see. If transcription was going way too fast, what do you think might be the worry? Sorry? So that could be, but what else might be an issue? So with, sorry, what?
Translation wouldn't keep up.
Okay, so what happens if translation doesn't keep up? Robert?
Could the mRNA bind with itself?
It could. Yep, that could definitely happen. Anybody else? Sorry, what?
Wouldn't it just stop?
Eventually it'll like go too fast and it'll like terminate. Yep. Anybody else? So what happens is, so RNA is actually a very fragile molecule, and it's single-stranded. So like Robert said, it will fold and it'll start, binding whatever, and it might protect itself. But single-stranded RNA is just going to get chewed up pretty easily. And so if transcription was going way too fast, you do run the risk that your mRNA gets degraded before the protein actually gets made. And so the cells have magically made it so that their translation and transcription rates are essentially, being paired and moving along with each other, so that doesn't happen. And it allows it to just be the most efficient with this processes. And so that's where we have termination. Oh, I guess. Okay, so for termination, so termination again happens at the stop codon. So once that moves into the A site, so termination now requires these different release factors. So either RF1 or RF2 will enter into the A site. And then the peptidotransferase activity, so the same activity that's linking the amino acid to the growing peptide chain, so that same activity will be activated once the release factors enter, but this time what it's going to do is essentially release the peptide from the ribosome. Then RF3 will enter, and RF3 entering triggers the release of RF1. And then we have another ribosome recycling factor, so that will enter, and that one is enters with the EFG GTP complex. So the EFG is still kind of playing that same role that it plays in the elongation, and with the GTP hydrolysis, it essentially will kick off the final RRF. And so at that same time, so IF3 will come in and bind to the 30S, and so IF3 can actually just come in and bind there even once they're already separated. It doesn't need to wait. So we showed you the initiation. That was in the case that the two subunits were bound, but it doesn't need to wait. It can just come in and then just get ready to go. All right, so here's a problem for you to think about. So how many GTP molecules would it take to synthesize a protein that has 100 amino acids in length? So things to think about. What steps involve GTP? And how many peptide bonds, if it's 100 amino acids, how many peptide bonds do you need to form? And then don't forget initiation and termination, that also involve GTP. So I want you to talk to your neighbors and see if you can come up with a solution to how many GTPs you would need to make 100 amino acid peptide. We'll give everybody else time. right, any ideas? I know Ari has an answer. What?
3?
For the whole thing?
33.
33? What? I'm hearing 33, 100, 3. Anybody else?
101. He said 101.
This guy over here has an answer. 400. All right, so why don't you guys start explaining to me how you got these answers? So, yeah, nobody?
So,
You good?
like in one instance, during translocation, there is a GTP hydrolysis.
During
And then
translocation?
it attaches to like EF-TU and then EFG, so that's 2 more GTPs, so that's 3 so far. And then there's another one in initiation where the GTP is hydrolyzed, so that's just 4 times 100, right?
You got all the GTPs correct, but you only initiate once and end once. There's only one termination and one initiation, one termination. So redo your math. You're very close. Almost. How many bonds? So there are 99 bonds. Right, and so he basically laid it out for us. So there's 1 for initiation. So how many GTPs? And then there's 2 for each bond, basically, and then 1 for termination. So how many for 100? Yep. So you get times for each bond, you get 2, and then you have the 1 for initiation, and then the 1 for termination. So that's 200. Yep. Yep.
How do you get 99 bonds?
So if you think about there's 100 amino acids, so let's start with there's just 3 amino acids and they all have to be linked together. There's just 2 bonds. Yeah? Okay. Okay? All right, so just like with transcription, so this time there are actually more antibiotics that affect translation. So here's just 4 of them. So streptomycin binds to the 30S subunit. So this interferes with the codon-anticodon recognition site. So you actually end up with a lot of mistranslation in this case. Tetracycline, again, binds to the 30S subunit and it blocks the amino acyl tRNA binding to the A site. Chloramphenicol inhibits the peptidyltransferase activity of the 23S ribosomal RNA. And erythromycin is another one that affects the 23S ribosomal RNA and interferes with translocation. So, translation is a very important part, right, for the cell, right? It's making everything essentially that the cell needs, and so this is a very key step. And so you'll notice there are lots and lots of antibiotics that try to take advantage of that. All right, so we'll just take a step back and look at the big picture again as a kind of a regroup around transcription and translation, and just kind of tying that all back into the stuff we learned about genes from the last chapter. So this is just a picture of a gene. So here is the DNA, right? So it's the double-stranded, it's DNA. This one actually encodes for 2 genes here. So there's gene A here and gene B here. Here's where we have the transcription start site. So again, this arrow here is usually what we use as a symbol for the promoter, and it's usually placed at the +1 nucleotide, so the start site. So this one again is remembering, so transcription is going to happen, it's going to come in and it's going to look for the polymerase is going to look for the promoter site, so marking off the -35 and the -10. So we'll have transcription that's going to start here, and so this is your mRNA that's being made. So you notice that it's specifically this length here right? So this is the terminator here. So it's not showing, there's no mRNA, doesn't include all of this sequence here, because that's where the start and the stop is for that. And then, so in the mRNA, so this one again, so we had 2 genes, right? So we essentially have 2 ribosome binding sites and 2 start codons. And so here, what we'll have, you'll often hear of the 5' untranslated region in these, and so these are the untranslated leader. And so within that is the ribosome binding site. And so you have the ribosome binding site, and here's your start codon. So this goes, and you get translation of this protein here. But here's the stop codon, right? So the picture is making it as the length. That's not really how that's going to look, but this is just to show you that this is where it's starting and stopping. But then again, this has 2 genes, right? So there's another start codon here, and you translate this protein. This protein is smaller. There's another stop codon. So there's 2 stop codons for each of these. Okay?
And the ribosome binding site is the Shine-Delgado sequence?
Yep, same thing. All right. So now we'll just talk about some protein modifications. So there's a lot of things that has to happen so it translates the protein, but then that's not the end of the story. So so it can be modified after translation in several different ways. So the N-formal group, so that can be removed from the methionine. So remember, the first one that comes in is that F-met that has that formal methionine on it. So that can be removed by methionine deformylase. And then in some cases, the protein doesn't even want that. It doesn't need that at all. And so it just needs it to start translation. So it'll remove the whole thing. In other cases, you can get acetyl groups or AMP to be attached. Some cases, there will be proteolytic cleavages that can either activate or inactivate a protein. So a lot of modification steps can happen after to get to the full active protein. One of the main things that's going to happen is protein folding. And so protein folding, so the final protein structure, is going to be determined by several things. So one, at the base, it's going to be the amino acid sequence, right? Whatever amino acids in whatever order is going to dictate what structure you get. But in some cases, the proteins can just fold by themselves based on that amino acid sequence. Like, it doesn't need any help, and it'll just know what to do. In other cases, it needs help folding into the right structure. And so there are other proteins called chaperones that will actually help do that. So there are kind of 2 of the major kind of categories that your book covers is these GRO-EL and GRO-ES. So these ones will refold denatured proteins, and it uses ATP. So this makes these kind of barrel-shaped ones, where the protein actually will go into here. So it'll go in here, and it'll actually go into that barrel, and that will actually help it refold. The DNA K, so this one doesn't actually form a ring. So this one, in this case here, the 2 different parts here will actually clamp onto it and bind on there. And that, again, will assist with peptide folding. So this is just kind of an overview of what this looks like. So we have, your book also talks about trigger factor. I'm not going to go over all the specific ones and how they all work. But so as you have the peptide coming out of the ribosome, this stuff can happen immediately, and so you can have the different chaperones come and attach to the different proteins. Some cases it'll just fold directly into its active form on its own. In some cases you can get passed through into some of the GRO-EL complexes. And then it'll get folded. So what else can also happen is that if it notices that there are mistakes or there are errors, what can happen is that it'll actually go into a protease, and it'll start chopping it up into the different, so you'll see here, it's the degraded proteins, the individual amino acids. So if you have a misfolded or just a mistranslated protein, it can also just chop it up. We'll talk about that now. So so for protein degradation, so you can imagine this in cases where both you have misfolding, but then also in the cases when the protein's done its job and the cell no longer needs it, the environment's changing, it needs something else. So proteins contain degradation signals, which we call degrons. So there's what we have the N-terminal rule. So the N-terminal amino acids correlate with its stability. And so the idea behind this is that depending on what amino acids you have at the N-terminal N, that dictates the half-life. And so some of them are more stable and some of them are not. So if you have these, you have a shorter 2-minute half-life, but if you have some of these, you'll get the longer half-life. So a lot of the proteases, you'll notice they're called CLP, so CLIP. So they'll have different, and we'll see some of the other names on the next slide. So CLIP-S recognizes the N-terminal amino acids and presents the protein to this CLIP-AP protease. Again, like we just saw in the diagram before, the abnormally folded proteins are recognized by the proteases, but in part because the hydrophobic regions become exposed. So when they're wrong, so the cytoplasm is you consider a wet environment, right? And so if the hydrophobic regions are sticking out, there's something wrong with that protein. And so the proteases will go and they'll recognize that, and it'll start chopping them up. Okay, so so all of these, you generally use ATP hydrolysis to help unfold the target protein prior to digestion. So again, these are generally these barrel-shaped things, right? So they have these places for the protein to go in, so it needs to unfold it to fit. So what you'll notice is that, so these are some of the examples like CLIP-AP, CLIP-XP, CLIP-YQ. So these actually have these different caps. So the X, the A, the B, the C, or whatever, these are different caps. And so you'll notice the name. So let's see. So CLIP-AP and CLIP-XP. So the P is the same base, but then it has a different cap. So this one has the A cap and this one has the X cap. And that'll just recognize different sequences. All right, so we talked about protein modifications and then what happens when you're done with the protein if you have the wrong signal. But now, if you remember, and this is especially true for gram-negative bacteria, right? The cell envelope contains proteins in it, but it has different regions in the cell envelope, right? So how do we get the proteins to the right locations? Yep, so that's definitely one of them, right? So these all require special export systems. And so the first one, again, we talked about this before, that's the SRP. And so that, again, will bind to these 15 to 30 amino acid sequences and target that so that's just to the first membrane, right? So that's to the cell membrane. And we'll just finish this up next time. So there's just like 2 or 3 more slides and we'll finish that up next time.